Hi, Šárka Lehtonen. You are a research director and the leader of the Human Brain Disease Modeling Group at the University of Eastern Finland. Could you briefly introduce your research group and some of its key focus areas?
Sure. I lead the multidisciplinary team that currently consists of 12 persons, one senior researcher who interacts closely with 2 postdoctoral fellows and 5 PhD candidates. We also have several students who are working on their master and the bachelor thesis, and continuously we welcome interns from Finland and internationally who we train in laboratory skills, mainly in using human-induced pluripotent stem cells, which actually serve also as a primary resource for building up human brain disease model for our research. My group focuses on utilizing the IPC lines and differentiating them to the cell type that allow us to study cellular and molecular mechanisms of synucleinopathies like Parkinson's disease and multisystem atrophy. We are especially interested in identifying the initial pathology events like alpha-synuclein aggregation and its propagation in the CNS, but also in a periphery autonomic system.
Okay, and what are some of the projects that you have going on?
So lately we have started 2 projects that relates to the synucleinopathies and the early events, but they tackle the problems from 2 different angles. So the first project investigates the newly discovered alpha-synuclein gene mutation found in a Finnish family with multigeneral history of atypical Parkinson's disease and is linked to the early onset Parkinsonism, meaning that the first patient actually has been diagnosed as early as age 36. And this mutation has been previously associated with atypical multisystem atrophy and a PD pathology based on the clinical, neuropathological and genetic findings. So what we are doing here is that we are using IPC lines that they obtained from those individuals, and we are using them to model midbrain. So we use the midbrain organoid slices and also the advanced technology or methodology like single-cell sequencing and spatial transcriptomics or electrophysiology or immunocytochemistry. In the second project that explore rather the body-first subtype of the synucleinopathy, where the pathology originates in the periphery, likely in the enteric nervous system, and spread later on only to the brain. And this subtype is associated with long prodromal phase before the motor symptoms appear, and it can be even the decades before actually the patient is diagnosed with the Parkinson's disease. And here we focus on the early disease mechanism, and we studied gut microbiota in alpha-synuclein pathology transfer, and using also the organoids or organoid slices, but this time they originate from the intestine, and we incorporate the enteric neurons into the organoids. We also use the gut-brain axis neuronal models, where we use the chip and place the enteric neurons on the one side of the chip and connect through the microchannels to brain side, which is represented here by dopaminergic neurons. So by studying these initial events in both of the projects that I mentioned, we hope to get insight into the neurodegeneration and its early stages.
Wow, sounds very interesting. And what kind of collaborations do you have both within the university and maybe beyond it?
Yeah, the collaboration, of course, are very much important because our projects are rather wide-ranged, and we do collaborate with neuroscientists nationally, but internationally as well, where we are exchanging the methodology expertise. We do collaborate also with clinical neurologists and neurosurgeons who are providing us the biosamples, like, for example, skin biopsy, that from which we can make the fibroblast cells and reprogram them to the induced pluripotent stem cells that we use later on. Also the blood or the brain tissues, which are collected after the deep brain stimulation surgery, which some of the PD patients are undergoing. I want to also name the pathologists who are basically providing us the post-mortem brains, and already from the mutation that I mentioned, 2 of the patients has died and has donated their brain for the research purposes. And of course, the bioinformaticians and the computational biologists are really needed to help us with analyzing the big omic data and also integrating some functional data. And at the end, I would also mention pharmaceutical company. As being a pharmacist, we also do collaborate with a pharmaceutical company. We evaluate the molecules in our human-based models that we established in my lab.
Whoa, such a great variety of collaborations. And what are some of the achievements of your research group that you are most proud of?
So the existence of my group dates back to 2021. And while big achievements are still ahead of us, I'm already incredibly proud of everything that we have accomplished thus far. So I'm proud of the entire team, their dedication, their commitment, the collaborative spirit, our high quality scientific work, strong motivation and passion to learn new methodology and set up human brain disease models. And of course, I'm proud of the networking and the collaboration that I mentioned, because these are really essential to increase the impact of the translation research that we are doing.
And talking about impact, what kind of impact do you wish your group's research would have in the next maybe 5 to 10 years?
Yeah, so in the regards of these 2 projects that I mentioned, I do expect that we might be successful in revealing some novel therapeutical targets for personalized treatments. So far, a rather small number of the individuals that were diagnosed with severe mutation in alpha-synuclein, and this way we could advance the precision medicine. In case of the second project, I would expect that the outcome of the study will enhance the early detection, enable early diagnosis, which could be possibly decades before the motor symptoms manifest, and improve the outcome through timely and targeted intervention. Overall, for all projects that we are carrying in my group, I hope that we might contribute and bring new tools and hopefully some disease-modifying therapies that might improve the lives of patients that are diagnosed with synucleinopathies.
Very impactful indeed. Then the most important question, why should researchers choose your research group for their postdoc? What makes your research group so amazing?
Yeah, this is really good question. And I think I would start maybe with saying that Finland has been again voted as the happiest country in the world. And Kuopio as a town is an excellent place to live, work, and raise a family. And I'm a great example of it. I came to this university to complete my master thesis as an exchange student, and it has been more than 20 years, and I'm still here. And the reason why I stay was very much welcoming and supporting environment in which I had the opportunity to work and grow as a neuroscientist. And this also reflects the atmosphere I try to set up in my group. So what makes my group very special is that we try to support and encourage one another. We exchange the thoughts on the daily basis. We try to be transparent. We try to create the environment that promotes the curiosity, resilience, and the teamwork, not only push the limits of our research, but also grow as people and as a group.
Wow, superb. Thank you so much, Šárka.
Thank you, Pirita, for this time.